All right. Good evening, everybody. Thank you for joining our latest Apiculture Online Hive Chat with NC State. My name is David Tarpey. I'm the Extension Apiculturist here at NC State University. And as usual, we're joined by members of the Department of Agriculture here in North Carolina and the Apiary Inspection Service, as well as others from our program. So thank you all for joining us on YouTube. We're live streaming on YouTube, or you're watching the recording afterwards. So make sure that you comment, like, subscribe to our channel so that we can keep these videos going and the discussion going about bees and beekeeping. We're going to, we have a lot to talk about tonight. This is a really important time of year, and there's a lot going on with bees and beekeeping. And so before we begin, I just want to make one last final announcement about our Bees Academy this upcoming weekend. So Saturday, October 2nd and Sunday, October 3rd in the Union County Extension Center in Monroe, North Carolina, is where we're going to have this two-day event where we're going to kind of go over an intermediate level of bees and beekeeping that's really ideally targeted for those at around the journeyman level, preparing for the journeyman level in the Master Beekeeper program. We like to think of it as kind of this very effective booster shot of your beginner bee school, where we go into more depth and a lot more breadth of these different subjects now that beekeepers have been keeping bees for 1, 2, 3 years or so. So it's really ideal for those beekeepers at that level. So we still have some seats left because it's a very large facility, which is really excellent. So there's more space left if anybody's interested. But we'll just go ahead and get right to it. The first segment that we always start out with is bees in season, which is in essence what your bees are doing right now and what you as the beekeeper should be doing in order to help them out. And so at this time, kind of late September, going into October, this is perhaps the most critical time of the year for the bees. The active season is starting to come to a close, again, depending on your location and the local climate and everything like that. But the things that the bees are really looking to do right now is making sure that they're storing away honey for the winter. Compared to up north, where winters are longer, down here, winters can be very short or even practically non-existent in different areas of the Southeast. But nonetheless, there's still a winter dearth when it comes to a lot of ample forage. And so they definitely need their stored honey. And so if you feel like they're going into winter fairly light, it can be very helpful to supplementally feed some sugar syrup right now to make sure that they get that into the comb so that they don't have to break cluster in order to get their food, in order to start raising brood early next spring and to generate heat through the winter. In doing so, if you are going to be feeding in the next kind of month or two, feeding a very thick sugar syrup, pretty much as thick as you can make it, about 2 parts sugar to 1 part water, is the best way for them to be able to process that and get it into honey form and stored into the comb so that they'll have it. It is certainly possible, and beekeepers do it, but ideally you want to avoid having to feed during the winter, because you can't really feed liquid syrup during the winter. It's really best to give other forms of feed. We have some previous videos on that if you want to go look those up. But it's best to be proactive and preventative in making sure that they're going into winter with enough honey stores. Another thing that the bees are doing this time of year is that because they're brooding down and the brood nest is kind of contracting, they're often backfilling that empty comb or the freed up space from less brood within the comb with pollen. So they tend to store more pollen going into winter and turning that into bee bread than they do during kind of the normal times of years, especially when there's a pollen flow and they can always go out and collect more. So they're kind of backfilling the brood nests with a bit more bee bread. So hopefully there are still some pollen sources out there for your bees. Check on the colony entrances as they're coming in and out to see if they're still carrying pollen loads on their hind legs. And then one last thing that can really be the death nail of a colony this time of year is if a queen goes bad this time of year. So make sure that you have a good queen laying a good brood pattern. Again, the brood nest is shrinking. That is, she's not laying as many eggs, but those that she is laying, you want to make sure that they're good, so that you're going into winter and through winter with a good queen coming out next spring. Of course, this is also the most critical time of year for mite control and monitoring your mites. So this is something that we've been hammering on in our Apiculture Online for the past couple months, of course. And so there's lots of recordings on this about how to monitor for your mites, different options for control, all of those types of things on our recordings. But I want to stress once again that you need to control those mites, make sure that they're under the threshold where they're going to really impact the health of your colony, which is shockingly low, a lot lower than what most people think. And you need to continuously monitor, even if you've already checked and monitored and the mite levels are low. Because as we talked about last month, the mite levels can go from low to very, very high just within a few weeks, depending on how many other colonies around you may be collapsing from the mites, and you get immigration from robbing and other sources of the mites coming into your colonies. So just because you've been doing the right thing doesn't mean necessarily that the mites are always going to be low if you checked them a couple weeks ago and they were low then. You really need to be on top of that just to make sure. So, not continual, but very frequent monitoring, ideally every time you go into your colony and you do one of those alcohol washes or do a sugar shake, just to make sure that the bees are not going to be parasitized too much, because right now or coming, in the next couple months, that's when they're going to be making these longer lived winter bees that are kind of big and fat and have lots of energy stores in them. And so if they're parasitized during development, then they're not going to be very well nourished, and therefore they're not going to live as long through the winter. And that's why mites can lead to winter mortality, because they just don't have the longevity. Those bees don't have the same longevity that they normally do. So one thing that you should be doing in preparation of going into the winter, it might be still a little bit early in many places in the region that may still have kind of a month or two before things are really kind of batten down the hatches. But there's an old adage in beekeeping that you want to take your winter losses in the fall. And so what that means, you want to reduce your hive size and unite colonies accordingly. And so by uniting colonies, I mean, it's going to be a lot easier to take two moderate or weak colonies, and rather than trying to nurse them through the winter and expecting them to make it, which they probably won't and both will die, it's better to unite those, or better yet, unite them onto a stronger colony, so that you go through with one strong colony, and then next spring you can split it. It'll have a much higher chance of survival, and then you'll be able to replace your fall losses by making splits from that strong overwintered colony. So using the generic newspaper method, where you put a newspaper in between the two colonies so that they don't mix and fight each other. The workers don't smell like each other. So that gives them a little time, because the newspaper is porous. It'll allow their hive odors to blend. And so by the time they chew through the paper, they will be much more readily accepted with each other, and you can unite those colonies and save those weaker colonies that probably might not make it on their own through the winter. It's just far better to unite them and come out with a strong colony next spring, rather than roll the dice on weaker colonies. And the same thing when it comes to kind of reducing the hive size. So a lot of times this time of year, as the brood nest is kind of shrinking, it frees up a lot of space. They don't necessarily always backfill it with everything. They might not have all of the frames filled with honey and everything else. You might have a lot of frames in here that are just kind of empty. And so it's better to get rid of all of those empty frames and knock this two-story colony down into one, and put, so that all the frames have as much food in them as possible, all the brood that they had goes in there, but you really kind of minimize the number of empty combs because they're not going to do anything through the winter dearth. I particularly favor, rather than just doing a 10 frame, knocking that down into 10 frames, I like to put those 10 frames into a stacked 5-frame nuke box. So, nukes are really, really terrific because the overwintering, even though it might be kind of a smaller hive, or at least not as wide, the stovepiping action of the heat and the thermal dynamics through the winter cluster can really be beneficial. And so if there's honey above the cluster, they start down here and then eventually they wind up here. And it's just, it can be a lot easier and better, both for the bees and the beekeeper, to overwinter vertically rather than horizontally when it comes to their different resources. So it's just something to think about. There's no kind of magic system here other than getting rid of those empty combs that really aren't going to do much for the bees through the off season, as long or short as it is. So again, one of the things that we as beekeepers need to be doing is thinking 1 to 2 brood cycles ahead and where the bees are going to be, and that is about 3 to 6 weeks from now, so that the things that you do now will help set them up 1 to 2 brood cycles from now so that they're going to be in a much better position, right? So what that means, of course, is that 1 to 2 brood cycles from now, they're going to be going fully into that overwintering mode. They're really going to be, all the winter bees are going to be emerging, or at least being reared at that time. So you want to prepare for that. You want to make sure that they're going into that less parasitized by mites, and make sure that they have plenty of resources to be able to rear those special bees. They're going to be brooding down and continue to brood down, as I was saying. And as our timely topic tonight, we're going to talk about how you can move hives during this time to good overwintering yards. So that's another thing that you can do, is take advantage of different locations to place your bees that have different, subtle different advantages of where they're located during different seasons. So with that, I'm going to check really quick to see if there's any questions here. I don't see any right now, but feel free to type in your questions on the YouTube channel as we continue to go through. And of course, we're always going to wind up the last 15 minutes or so is just an open question and answer session, so you can save them then as well. So with that, I'm just going to then launch right into our timely topic. And as I just alluded to, the timely topic today is moving hives to improve overwintering success. And this topic came up because a couple webinars ago, there was a question or a comment on the YouTube channel that that particular beekeeper was taught in beginner bee school that moving one's hives is really kind of dangerous or problematic for bees, and so it should be avoided. And I was really kind of struck by that, but I didn't really go into it very much. And there is some empirical evidence for that. This is actually a study that we had done and published several years ago, where we were looking at bees that were either stationary, that is, they weren't moving, or migratories, that they were being moved every 3 weeks in and out of different agricultural settings. And then we took those bees and we emerged them in an incubator, and we put them in very highly controlled conditions, and we looked at their survival. And what we found is that the bees that were not moved, that were stationary, versus those that were stationary, the stationary ones lived about a day longer than the migratory bees, which was in fact statistically significant, but is really only about a 5% or so reduction in lifespan. So it's a modest but perhaps biologically relevant effect. But in that same study, what we found was that the migratory treatment, moving bees around overall, didn't affect colony size, that is the adult population or the amount of brood, which in the end is the most important thing when it comes to beekeepers. So that subtle effect was not translated at the whole colony effect. But more importantly, we measured the oxidative stress and like the physiological kind of how robust the bees were. And in essence, what we found is that it wasn't about whether or not the hives were moved or were stationary. What was important is that if they were in a good location with a good pollen and nectar flow, they tended to be much healthier in these measures. But if they were in an area where there was dearth, they had lower scores on this. So in essence, what that means that the stationary bees did better than the migratory bees in the spring, but in the summer, the migratory bees did better than the stationary bees because the migratory bees were being moved in and out of agricultural settings where there was still something to eat, where the stationary bees were in kind of a food desert because nothing was blooming in that area. So what really matters is location. And so, you know, moving hives can be a pain, and it can put a temporary stress on your bees. But overall, if you're moving to a better location, then that is better for the bees in the end. So that's just something to keep in mind. And so choosing apiary locations is something that a lot of beekeepers learn about in beginner bee school, but most of the time they're just choosing the location based on convenience. They're doing it in their backyard, or they're doing it on their farm because that's where, you know, they have, that's the best place to put the bees. But if you really want to take your beekeeping to the next level, you want to think about other locations, and in fact having multiple locations for your bees and move them around from place to place. We all know that, you know, the standard thing, you want to have a nice good water source that's close by, you want to have a windbreak, you want to have, you know, morning sun, afternoon shade, or at least, you know, something where it's not full shade at all. But a good location in the summer can be subtly different from a good location in the winter and vice versa, right? You know, in the summer, it's good to kind of keep the bees cool, but you don't necessarily want that in the winter. It's good to have them in full sun. You want to have a good windbreak so that they don't get too chilled, but you want good airflow in the summer, right? You always want water nearby in the summer or in the winter, but it's important that not all locations are good for all seasons. And so there really is no kind of one ideal location. And so moving your bees from your kind of summer yards to an overwintering yard and then back somewhere else in the spring can really help with your overwintering success in kind of, again, subtle but important ways. Now, if the real difference of moving your hive or hives in your backyard from a kind of shady spot during the summer where they might stay cool, but then moving them over to the other side of the yard, you know, 100 feet away or something like that, where they're in full sun during the winter, that can be kind of a difficult thing. Because if you just move them 100 feet, especially during the day, then all of the foraging bees that are still out can get lost and they won't make it back. So there's two ways to move your hives just kind of within an apiary or within a location. The best way to prevent losing those foragers is to temporarily move the bees at least 3 miles away for about a week, so those foragers reorient and learn the new sighting or the new site, and then move them back to the other side of the yard, and then they'll have to relearn yet again. And so when you move them at night, you're not going to lose any of the foragers, and they're not going to be confused by being in different sides of the yard. The other way to do it is incrementally, where you can move the hive, you know, maybe kind of 2 or 3 feet every single day, ideally in the middle of the day, not at night, but in the middle of the day, so that eventually, they don't get too lost, and they'll able to keep up. But that can take obviously several days depending on how far you have to go. Now, if you want to move between apiaries, and again, really urge you to kind of locate, scout out, and ask around to different people in the community who are looking for places to put their bees. You can find these alternative apiary locations. And in doing so, what you want to do is you only want to move your hives at night. You want to screen off the entrance so that they still have airflow, and that they have ventilation so that they don't suffocate, but clearly they don't come out and be really defensive during this very disruptive move. You want to make sure you use hive staples or straps, something to secure all of the different hive bodies together. Many a beekeeper says, oh, well, they have a lot of propolis and it's very gummy, so we don't need that. And they regret that they didn't use some other reinforcement to make sure that those boxes didn't come apart, because that can be a real mess during a move. So using something to secure all of your hive equipment together. And then, of course, practice good ergonomics and lifting with your legs, and ideally with a partner or somebody else, a helping hand with a strong back, so that you can keep yourself safe as well. But, moving the hives during the evening or during the night, so that you can take the entire colony to the new location, open it up. Don't forget to remove that entrance screen so that the bees can come out the next morning, reorient to their new locality, and then they'll be able to set up shop there immediately. And so again, this is a subtly or a less, it's not used very much by your kind of backyard hobbyist beekeeper, but it's something that I think can definitely help and can improve overwintering success depending on where they move. So, my final thoughts on all of this, and I'd like to hear others on our Zoom channel here of what they think as well. But, different locations vary in how appropriate they are in these different seasons. Again, some locations are great spring yards because they have a great local nectar flow or something like that. But then once that stops, it can be a horrible summer yard. It can be a very bad overwintering yard. So not all locations are ideal all times of the year. And so, optimizing your locations of your hives and moving them from time to time, from site to site, can really help to optimize their nutrition. And the more, if you think about it, the more that you can move your hives into areas that have good flows, rather than dearths, that's going to save a lot of money in the long run, where you're not going to have to feed supplementally. And it's also going to increase the survival of the colonies, because they're going to be nutritionally better off and therefore survive better. And so, moving hives can be one of those underutilized tools in our toolkit that a lot of people don't necessarily think about. Other thoughts from the panel on moving hives? Go ahead, Louis.
Hey, David. I just wanted to say, I think as far as overwintering, I gotta say, there's nothing more important than healthy bees going into winter. If your bees are sick going into winter, they're not going to overwinter very well somewhere else. They have to be healthy. And that goes for people like to insulate their hives or whatever for the winter. And it's just, in my opinion, garbage in, garbage out. If your bees are healthy, no problem. If your bees are not healthy, it's going to be hard to get them through. So
yep,
double down on those, on those virus checks. The varroa checks. I am starting to get calls from people who are removing their treatments and finding that they still have mites. Yep. And can't stress that enough. Just because you did something doesn't mean it.
No, you're exactly right. And so all else being equal, right? So you're exactly right.
Yep.
That, this is moving the hives to a different location isn't going to save a lack of control over varroa or anything like that. Yeah. So you're exactly right. So that is the first, second, and third priority. Don, did you have a comment?
I wouldn't have said anything more than what Lewis already mentioned. Yeah, if they're healthy going in, it goes a long way to having them come back out in the spring.
Yeah, and I think a lot of people in the Southeast think, oh well, we don't really have much of a winter, so they're not raising a lot of winter bees. So it diminishes its importance. But I very much disagree with that. That it really is critical that those developing winter bees are not parasitized during this time frame. But we don't really know exactly kind of when they are necessarily reared, but we know it's kind of within these few months here during this season.
Yeah, they need to make it through till March.
Right. That's a long time for bees that usually only live about 6 weeks, right? So important. Great. Let me quick check the comment. There's a comment here that suggests using flashlights with red lenses when moving at night. That is fantastic because bees cannot see red, and so that helps us see, but it doesn't alarm the bees as much or make them as flighty necessarily. If they sometimes they can take flight when they see the light at night, and so that can help. Doesn't help me very much. I'm red-green colorblind, so I can't see the the red flashlight very much. But that's still very good advice to be able to have our cake and eat it too during those times. All right, so then I'm going to go ahead and share our screen again, and we'll go to our final thought, or we'll turn it over to our third segment here. I'm going to introduce our guest speaker today, or not guest speaker, our guest in Apiculture Online, who is my counterpart in Indiana at Purdue University, Dr. Brock Harper. Brock is, well, he got his PhD in Canada, and he moved down here and started at Purdue in 2019. So he's kind of new on the scene, but he brings with him a lot of excellent work, and he's done a lot already in a very short career so far. And so he's really well recognized as a rising star in apiculture and bee biology. And he's taking over a very well-established program at Purdue and just taking that to the next level. So I'm really, really happy to introduce Brock, and happy to have you here.
Hey, thanks for that. Nice to be here.
Yeah. So so I know that you have research and extension mandates in your program, as many of us do. And so tell us a little bit about some of the research that you have. I know that you're coming with already kind of a research paradigm using a lot of genomic tools and trying to understand the underlying kind of genetic architecture of bees and what that means for bees and beekeeping. But as your program grows and develops, you're going in some other directions as well. So why don't you tell us a little bit about those?
Yeah, absolutely. So I've really been trained as an evolutionary biologist. So I'm, and with most of my interest in honeybees and social insects in general. So a lot of my extension is kind of informed by the research I do. So as a lab, we're very interested in understanding bees as an evolutionary model organism. So we start from the ground up. I like to say we kind of start at the baseline genomic level. We want to know what mutations are out there in the honeybee genome. We want to know how they got there, and we want to know kind of their distribution in space and through time. From that understanding, we then like to make connections between genetic variation and function. So when there are mutations that are, we'll say, functional in loose quotation marks, how are they impacting different phenotypes, different traits of interest in individual bees within a colony, and then between colonies? So how do we make those connections? How are mutations doing that? And then, of course, what are the mechanisms? What's a good
example of that? So what's a good example of a mutation that you found that has a benefit to bees and colonies?
Yeah, that's a fun question. So there's a long history of bee genetics, especially in the United States. You mentioned the lab that I took over, Greg Hunt. So Greg did a lot of work on defense response in honeybees. We know a lot about the genetics there. I've spent a lot of my time looking at different forms of social immunity and the genetics therein. So hygienic behavior is a very popular trait for bacterial resistance in bee colonies, and beekeepers in the part of Canada that I was working in had a long history of selecting on hygienic stocks. So we've identified some mutations, some genes in the genome that are associated with that. And we're doing a little bit of work to figure out how exactly they operate. So what makes one single bee, what causes single bees to be slightly more hygienic, and then how that integrates into the colony level. So that's really been a trait of interest right now.
Yeah, exactly. And I think, a lot of beekeepers are familiar with that, but it's also up until now been measured at the colony level, right? The way that it's done is you freeze a section of brood, you come back a day later, see how much of it has been cleaned out. That's your measure of hygienic behavior. But what you're talking about is, well, was it one bee that was just super anal and cleaned it all out, or was it everybody pitching in? And what are the genes that control for those traits? And if we can favor those, then we can have, way more of those super anal bees rather than, just kind of doing it in this crude way. We can actually select from the bottom up rather than the top down. Is that right?
Pretty much, yeah. So a lot of this, a lot of the connections that we're trying to make between genetics and phenotypes and traits of interest are so that we can select bees using genetics instead of phenotypes. So you've done a hygienic behavior test. I've done far too many hygienic behavior tests. They're awful. It's a lot of manual labor. So it would be kind of nice if we could, through inexpensive genome sequencing, make some predictions about the breeding values of queens, the breeding values of their drones, and the likelihood that a colony is going to express a trait of interest. Not all traits are great for that. Hygienic behavior is a great one because we know that there is a genetic component to it. Some traits are pretty messy. I would love to be able to predict honey production, but there's so much environmental noise there that I very much doubt we'll get to a point where we can do that.
That's right. Yeah, there's no gene for making honey, right? It's
well, we know there's genes for collecting nectar, right? So great work by Rob Page on that one. But the environmental noise that comes into that, 1 year, yes, on average, a gene predicts that they'll collect a little bit more, but one bad year.
Exactly. Yeah. So and there's lots of genes. It's not just one gene, right? It's
correct.
It's lots of different things that go into that. So, well, what are some other another big one that everybody's always wanting is some sort of a disease, and especially varroa resistance, or at least tolerance, right? And you inherited that really very famous Indiana ankle biter strains, right, of bees. So what's new with or for our viewers, go ahead and explain kind of what that is and where that program is going.
Yeah, sure thing. There's two traits that we're really pushing on now. That's one of them, mite biting. The other one, which maybe we'll talk about, is some stuff with males. But so the breeding program that I inherited has been going for almost 20 years now. It started and is still going through Purdue. It's run by Crispin Given and formerly Greg Hunt, and now myself. Crispin and Greg started by essentially selecting colonies that survived and colonies that tended to have lower levels of varroa mites. They did pretty well for the first few years, but they started to notice that some of the mites that they were collecting had damage on them. Legs were missing, parts of their cuticle had been chipped, looked like they'd been chipped off. That was quite curious. There's a whole host of reasons why that can happen. But they noticed it and they decided, well, let's see if we can select on this. Maybe it's bee related. They did exactly that. They selected on mite biting behavior, damage on mites, and they found that it increased through time as they selected on it, which implies that it's got some genetic component to it. So that they started at about 5% of the mites in a given colony had evidence of damage, and now it hovers around 30 to 40% of mites. There's some asterisks around that estimate. We find that the highest mite biting colonies don't tend to have enough mites for us to believe the counts that we
Yeah,
get.
that's
So,
true. That's cat. It's a self correcting
yeah.
measure. That's a good point. Yeah.
Yeah. So we find that most of the time mite loads correlate with mite biting. So higher mite biting, lower mite loads. So as you just said, there's going to be a cap at the top there. So we find about 30 to 40% of our mites on a bottom board have evidence of damage, and we've been trying to figure out what's causing this and how we can incorporate it into kind of a genetic approach to understand what's happening on an individual level and how we can incorporate it into breeding programs.
Yeah. So actually, there's something a little analogous to the hygienic behavior. Is it like one super anal, bee with huge mandibles going around and doing all of this damage, or is it everybody?
Yeah.
And so the answer to that question bodes a lot of how you're going to be able to foster that trait, right, and keep it around.
The big challenge with this trait is we haven't yet come up with a good individual assay. So what's wonderful about working with honeybees is when you open up a colony, you have tens of thousands of individual workers, which share half of their genetic material with their mom, and they have one of 17 some odd patrolines from dad. So they're genetically pretty similar, but divergent enough that you can test them all. They've all been raised in a common environment. Yep. It's really a wonderful model if you can get those individual tests. So things like defense response are easy. I can get an individual honeybee to sting me quite readily.
Right.
I can watch bees pull out larvae from a hygienic behavior test. Mite biting is hard.
You can't just give them a mite and then start chewing on it, right? Yeah.
Yeah, so there's a few videos floating around of folks doing that. It looks a lot easier than it is. We've had very little luck in those types of assays.
Yeah.
There's some current hypothesis is that mite biting is just aggressive grooming. So everyone's seen their bees groom themselves. Mite biting is kind of the extreme form of that. So we've tried some grooming assays. Those seem to work pretty well. Need to do some more work connecting grooming at the individual level to mite biting at a colony level.
Yeah.
But yeah, it's a fun, difficult trait to work with.
Yeah. Well, the thing that I've always really appreciated about that trait is because it goes back to the reason why varroa mite is not a problem with the original bee host, that is Apis cerana, the Asian honeybee.
Yeah.
Right?
Yeah.
It's not a problem. It's a minor nuisance.
I
Why?
think there's 4, yeah.
Because
you'll get like an average
they
of
have
4 in
bigger
a cell.
jaws. Yeah. And whenever they come across a varroa mite, they just kind of puncture it and kill it, and then it's not a big deal. Our bees are just slightly smaller, and they just have slightly less powerful jaws where they can't really do that as effectively. But again, if you can breed for that trait because it is genetically controlled, then that bodes really, really well.
Yeah. So there's some fun work by Hong Mei Lee Barley looking at exactly the structure of mandibles in mite biting bees. So we've been collaborating with her on that project, and she is finding some neat differences in the structure of the mandibles of mites that actually are biting. We're not yet clear on whether or not that's there's some honing going on. They bite mites and the mandibles get honed a little bit, or if there's some genetic thing going on that they have a different genetic background and their mandibles are a little sharper.
Yeah.
So some fun work coming out of Hong Mei's lab on that one.
Very cool.
But yeah, it is a neat behavior. It was reported for the first time in Apis cerana in '93, and pretty soon after in Apis mellifera, but at a much lower level.
Right. Well, this program's been going on for a while now, as you mentioned, and Crispin has been doing great work and keeping the lines going and doing all the selection and everything. But on top of that, you've built up a real community of beekeepers who annually take a pilgrimage to Purdue University and do their II there, and there's kind of a breeding club of this trade and being disseminated all across the nation and stuff. So tell us a little bit about that.
Gosh, yeah. So we work very closely with the IQBA, the Indiana Queen Breeders Association, and the HHBBC, the Heartland Bee Breeders Cooperative. I hope I didn't get that wrong. I don't think I did. Both organizations work very closely with Purdue to breed for this behavior and others of interest. So they, as you say, make the pilgrimage to Purdue to work with us on insemination. And we disseminate some of the stocks that we've produced out of the yard. We take our best and we inseminate them and spread them out with those beekeepers, and they have the opportunity to do the same. They can bring some of their best stocks to us, we'll inseminate them, or they can learn how to inseminate them with us and take them away. The last I looked at the results, we are hitting about 10 states with our queens.
That's great.
So it's pretty cool. And it's when I started, Greg Hunt told me that Brock with Extension, the beekeepers really kind of, he said they do it for you. And I think what he meant by that is they really let you know what they want and you just have to listen. So with this project, I've really just kind of listened and tried to incorporate a little bit about what I do along the way. So they've done a remarkable job in disseminating these stocks.
And just to be very clear, in case any of our viewers here are thinking, oh, I want to be able to go into a magazine and buy some of these. Yeah. That's not how it's done. You don't sell them, right? You make it available, but the beekeepers have to they have to be active citizens in this venture, right? It's not like you can just buy a breeder queen and then graft off of it, or, no. They have to make them, right? They make them with you, with the stock, but it's not that you're just kind of selling them and it's out there commercially.
Correct. We are not commercial bee breeders. And nor are, well, some of the members of the IQBA are commercial bee breeders, but largely, yes, you have to be a member of this community to secure the stocks. You have to contribute in some way, both financially and with some personal stakes as well. Yeah.
No, exactly. But I mean, I really like that though, because it is a kind of a grassroots thing, and a lot of times that makes it a lot more sustainable rather than because there's been lots of different stocks over the years, right? Lots of different traits that people have selected for, and a lot of times, and maybe you can answer this, I'm not sure if the ankle biting or this aggressive grooming behavior is recessive and disappears over generations, or like it has to be maintained, or is it something that is kind of dominant and tends to stick around once they have it?
So yeah, recessive, and either way, if it's a recessive or dominant, it can dwindle in the population just based on gene flow and frequency in the population of interest. So with mite biting behavior, I actually did this experiment when I started, and Crispin, unintentionally, I did this experiment.
Some of the best experiments, right? Always.
Yeah, I brought in some for a different experiment. We've been doing some stock comparisons in common garden apiaries, and I brought in some stocks that I will not name that do not have mite biting behavior. And we, in this experimental yard, we actually ended up having higher than usual losses of all of our colonies because of varroa mites in the other stocks relative to ours. As a result, there was some requeening of our mite biting stock. And there were 3 requeening events that year. We measured hygienic behavior, mite biting behavior 3 times over that year. In the first round of testing, our mite biting colonies were as we expected, between 30 and 40%. The next round with the hybrid daughters, it dropped about 50%. The next round after that, it dropped a little bit more, and then the next, so there were actually 3. So it definitely gets diluted, but as does any trait based on gene flow.
Yeah. Well, I mean that's too bad, but that's just the reality of this system, right? I mean, I think that's why it's not easy. That's why it's this kind of constant push, of the rock up the hill. Yeah. And you got to stay on top of it. And I would argue that that's true for pretty much any mite control measure, right? That's always going to be that. But what's great about this is that it is something that's always there. Once you have those genetics in the colony, it's there. It's not like you have to, it's a passive thing after you've done the heavy work up front, right?
Correct.
That
Yeah.
hopefully makes any other kind of control or mitigating strategies down the road a little lighter. It makes the bar a little bit lower, right? And that's the whole point of it.
Yeah, the way I look at this, if we're going to use cultural controls of management, and here cultural would be you're using a resistant stock of some type, you still have to do your typical management. You still have to monitor for mites.
Yep.
And if you're interested in a phenotype, a trait of interest in that stock, you still have to test for it.
That's right.
No animal breeder, no animal manager, no farmer is not going to test their stocks, and we just have to realize that we need to do that. So it's unfortunate. Beekeeping isn't a set it and forget it practice, unfortunately.
Oh.
Well, perhaps fortunately.
Here.
Yeah.
Yeah, and so sometimes, as you were mentioning before, what do you measure, right? It has to be quick, cheap, easy, and very reliable, right? And so that if you do it and then I do it, we come up with the same measure across states, across time. That can be the challenge when it comes to bees, right? Because so many other things can affect that measure. Just, you have a heavy winter, we don't, we don't really have a broodless period for the most part in many places of the Southeast, whereas you guys probably go broodless, 3 months out of the year or something, so like all of these different things can affect those different measures. And so that can be a real challenge for sure.
Absolutely. Yeah. And actually, so this is even a challenge with mite biting. So we've started expanding the breeding program a little bit. And I'm doing it. It's baby steps to start. We want to see if we can select it in other apiaries in other states. One of the first challenges that hit us, even prior to starting the baby steps, was measuring the trait, as you said. I can give 2 different technicians the same slide of mites, 10 mites. I know how many have damage on them. They don't, and I could get 2 very different answers.
Yeah.
So we've known that challenge for quite some time. Where we're trying to solve it now with a little bit of clever engineering. Purdue, since moving here, I've learned this is a fantastic engineering school.
Yeah.
Canadian, so forgive me.
You're about to say machine learning, aren't you?
I am about to use the words machine learning. Yes. Yeah. So we've been doing some image analysis.
That's great.
And yeah, it's pretty cool. I think it was most exciting to see how effectively the algorithms beat the people.
Well, that's humbling, right? When you have Big Blue or whatever those computers are called that are smarter than us, right?
Yeah.
There was a really great comment / question on the YouTube channel about this, and saying that how does the Purdue program differ from the Russian program or like the VSH Paul line or some of these other kind of varroa tolerant, varroa resistant stocks that are out there? How are they similar? How are they different?
Oh gosh, there's a few different ways to answer that, I guess. I guess first and foremost, it's different phenotypes. VSH is a very distinct phenotype. VSH is hygienic behavior specific to varroa infested cells. And the Russian line is a distinct breeding program put together by the, I'm going to get the name wrong, the Russian Bee Breeders Association.
Yeah, the Russian Bee Breeders, but it was originally imported from Russia, hence
Correct.
the name.
Correct.
So it wasn't really a trade, it was just they were keeping track of the lineage.
A lineage, yeah, exactly. And they seem to have a little bit of varroa resistance. So I guess first and foremost traits, breeding practices. We track pedigrees pretty vigorously in our program. The incorporation of genomic information is certainly different in ours. I guess the best way to answer it is really it's just a different trait.
Well, one thing that they all have in common is that they all have some higher degree of hygienic behavior, right?
I have not, since I've been in the lab, I haven't tested for VSH or hygiene in our stocks.
Oh, okay,
Crispin,
okay.
yeah, in the past, Crispin has, and I'd have to chat with him a little bit more about that, but I believe that sometimes our colonies can have a little bit of VSH in them.
Okay.
Yeah.
Well, yeah. So I know that the Russian stocks also are very hygienic as well. But has anybody tested for mite damage in VSH or in Russians?
Yeah, again, this Hong Mei's done a fun study where she was comparing typical commercial with feral and mite biter.
Okay.
And there was no, the typical commercial, I believe, were from Georgia or California. And
So
they all had
presumably Italian.
Italian in very loose quotes, yeah. And they had pretty low mite biting. I haven't seen one in Russian yet. I do know that African subspecies tend to have a little bit more biting in them.
Okay, that's interesting.
Yeah.
Interesting. So I didn't realize we just kept the conversation going. I'm just going to open this up to any and all other questions. So please go ahead and type in your questions on our YouTube channel, and I'll be sure to ask our guest or anybody else on the call. It can be any general questions or pertinent to Brock and his program and everything we've been talking about. Who's in your lab? What students do you have that are starting out, and what kind of projects are they investigating?
That's a really good question. I love talking about my students and their projects.
Because we all know that we're not important. It's the students within these programs that are the ones that actually have all the fun. So
yeah, I edit papers and write grants now. Yeah. Yeah. So as I said, we've got these kind of three parts, right? We're interested in genetics and what's out there, function, and making the connection of those to fitness. So students do bits and pieces of all of those. I have 3 master's students right now. I have Maddie Carpenter. Maddie's been doing this really fun examination of the genetics of bees across the United States. So we've got most of the commercial lines that beekeepers have available to them. She's done some collections of feral bees across different parts of the United States. My wonderful collaborator, Margarita Lopez Uribe at Penn State, is also working on this. So we've got some fun stocks from Pennsylvania. We've sequenced genomes of all of those, and we're looking at genetic diversity, divergence among all of them. So essentially trying to understand how similar or different they are. The other kind of fun thing that she's added on there is trying to figure out where they come from. So right. No honeybees in the United States are not native. They've been imported for 400 some odd years. Another fun paper by Maddie that everyone should read is actually on the history of those importations.
Yep.
So we're trying to see what's still here. So if we look at the genome of go to the Russian bee, do we actually see Russian genetics in their mitochondria or in their entire genome? Yeah. So Maddie's got kind of a fun data set there. So moving up, so that she's kind of helping us understand what's out there, the genes that, the mutations that we'd expect. My PhD student, Garrett, is looking more functionally. He's trying to understand how mutations across the genome impact male fitness. The fun thing about males, of course, is that they're haploid. So their genetics are just really nice to work with.
Yeah, very clean and simple. Yeah, exactly.
Yeah. So Garrett's been doing this fun bit of work where he's tracking male honeybees using RFID tags for their entire lifespan, collecting them at individual time points, estimating measures of reproductive fitness. So looking at sperm quality and quantity. This might sound similar to a paper by Metz and Tarpey in 2020. Adding in some flight data. And then what's cool is he's got different lines of bees for each of these as well. So all the lines that Maddie sequenced, he's got reproductive data for.
Oh, so they're well, not to give anything away, but to see if there may be differences in flight duration or timing of the day or something like that across different stocks. Italians might fly earlier than Russians or something like that.
Yeah, exactly. And then how does that link up with their reproductive quality at that time? Right, so we've let's say we collect them at day 14 when they should be sexually mature, or at least have sperm at the end of their endophallus. What was their flight data, or what was their flight response up until that point? And if they have low sperm at that time period, did they fly a little bit differently than someone who has high sperm? And then linking all of that up with the genetics. So Garrett's got kind of a fun experiment that he's been working away at.
Yeah, no, it's very cool.
Yeah.
We do have a question
Oops.
going back to Maddie's because there's about a 15, 30-second delay between our conversation and the YouTube. So there was a question about Maddie's project and some of the genetics from the cradle of the forest, which are some of those feral bees that were collected in Western North Carolina. And you were saying offline beforehand that there were some interesting findings in that. Are you willing to share?
Yeah, sure. So we were kindly provided with some feral bees from North Carolina, the cradle of
the forest, yeah.
Of the forest. Thank you. And they turned out to be pretty neat. So I can't tell you too much, but I can say they have there's definitely some Italian in their genomes, which we would expect. But they also have some markers that we kind of associate with very old Apis mellifera importations. Cradle of forestry. Thank you, Lewis. Yeah, so it looks like some of the colonies out there are very old indeed, or at least share, and what I should say is they share ancestry with some of the older importations in the United
original
States.
Importations, the German black bee.
Well, I don't.
Mellifera.
It's hard to say if it's original, but it's certainly old.
Okay.
Yeah, because there's just been so many importations into this country.
Yeah, that's true.
Yeah, so they turned out to be really neat. We're still looking deeply at them, but for now, that's I think one of the most interesting parts. Is there's certainly some commercial in there, but there's some neat things as well.
Right. Yeah, it shows that there is hybridization, but it is cool to see these kind of historical echoes. And thanks to Michael Elliott, who has been the shepherd of those and is the one who's provided those. In no way did I intend to take any credit there that we were providing them, but he's been doing a great job up there. And so thanks to him for that, and to you for running them.
That's actually been one of the most rewarding parts of this project. And we're launching it in a larger scale very soon. So getting samples from beekeepers that they think are interesting has just been so much fun. We get vials of bees, we grind them up, we send them off for sequencing, and then we can tell you a little bit about their ancestry. Yeah. We've just recently started a larger scale project where I'm hoping to do that nationwide, where beekeepers can send me samples, and then our partner company can grind them up and get the genomes for us. So keep an eye out for that. We're hoping to launch it soon.
Well, I know a lot of people are really interested in that, and hopefully you'll get deluged with samples.
I hope so.
A good problem to have of having too many. So for us doing science, more data is almost always better. So it's just the logistical constraints of doing something with them, right, and the costs of it. So
I have two students who, if I don't talk about, I'll feel terrible.
Please do. Yes.
We were talking about connecting genetics to phenotypes, and one of the biggest challenges in honeybees in doing that is that they're a non-model model organism. We can do a lot with them, but we cannot do what a fly geneticist does with a fly. I can't put genes inside of them very effectively and see what happens, or I can't remove genes and see what happens. We're getting to that point. I think there's always going to be hurdles with that in bees. So my graduate student, Stephanie Hathaway, has been helping us develop this fun system where we can put a honeybee gene in a fly, turn it on in the relevant tissue at the relative life history stage, and see what happens in that fly. So when she's working on royal jellies actually right now, when we turn on royal jellies in the Drosophila brain, when they're adults, what happens to gene expression in the rest of the brain? Does that brain become nurse-like, or does nothing happen?
Right.
So we're hoping that it's kind of a fun system for us to begin teasing apart the functional effects of some of these candidates that we get.
No, very cool. And the last student, you said the fourth
student?
So
Riley Schultz just started. She's doing she's working on honeybee males as well. While we were kind of dissecting and working with male genomes, I dove pretty deeply into the older papers and realized that male honeybees are actually not haploid. Did you know this?
No. Well, maybe. I think I know what you're talking about, but
yeah.
Please explain.
They're mostly haploid. That is, they mostly, they're the result of unfertilized egg. But if you look at their tissues, all of their tissues are not just, they don't all have one complement of chromosomes from mom. Some of their tissues actually have two complements. And it's the result of what's called endoreplication. So the genome duplicates itself. So some of those cells actually have two sets, but they're two clonal sets. Most notably is the muscle tissue of the male. The thought of that is that by duplicating the chromosomes in there, the hypothesis, I should say, the reason we think that happens is to increase metabolic output. It kind of helps them fly.
Right.
Dosage
That's not what I
compensation.
was thinking. I was thinking
Okay.
something totally different. So that's really cool. I didn't know that, but that makes a lot of sense and really, really neat.
Yeah, so Riley's doing this really kind of fun project where she's looking at how that influences mutation rates in those parts of the genome and how that influences gene expression in those parts of the genome. So are males actually duplicating their muscle cells, the genomes of their muscle cells, so that they can get more output for flight?
Right.
So that's kind of going to be a little bit of a fun project for us.
Well, it just underscores just how amazingly fascinating bees are. And as much as we think we know, we're still just scratching the surface and just staring in the face of ignorance of all of this stuff. But very, very cool. Brock, just great to have you. Great work that you're doing. Keep it up. You've accomplished so much in a very short time span. So I'm really looking forward to your continued growth and success. So
this is really fun. Thank you.
Thanks everybody for joining. Thanks again to Don and Lewis for joining on the Zoom call. And everybody have a great evening. Thanks again.
Thanks all. Take care.